Hello students and my dear friends, myself Madhu Vancha. In this session, we'll see with the introduction to microbiology. Learning objectives. After completion of this session, we should able to define what is microbiology and define the various organisms, which means like what is meant by bacteria, what is meant by virus, fungi and parasite, and distinguish between bacteria, virus, fungi, and parasites, and understand the scope of microbiology. What is meant by microbiology? Microbiology, it is the study of organisms which are usually invisible to naked eye. Invisible to naked eye means which we cannot see with our naked eyes. So how we can see them? So to visualize the microorganisms, we need an equipment called a microscope. With the help of the microscope only, we can see the microorganisms. So microbiology means it is the study of organisms which are usually invisible to naked eye, which means it is the study of organisms, microorganisms. It is the study of microorganisms. To see the word microbiology in the Greek, micro means small, bios means life, and logy means study. So which means it is the study of small life forms. So small life forms are nothing but microorganisms. And the term microbiology was given by scientist Louis Pasteur. It was coined by scientist Louis Pasteur. Louis Pasteur is considered as father or founder of modern microbiology. Then, what is meant by medical microbiology? So, what we deal in the medical microbiology? So, medical microbiology, it deals with the causative agent of disease or infection, which means what are the bacteria they are causing infections, what are the viruses causing infections, okay, and how they are entering into our body, and after entering into the body, how they are causing the disease, like what are the toxins they are producing, and what are the enzymes they are producing, how the toxins and enzymes they contribute for the diagnosis, and how the toxins and enzymes contribute for the a disease, and laboratory diagnosis, prevention, and treatment we'll see in the medical microbiology. So it is the medical microbiology deals with the study of organisms and their role in the human health and diseases. So it also deals with the diagnosis, treatment, and prevention of various infectious disease. So based upon the morphological characteristics, microorganisms, they are mainly classified into 4 groups. First one is bacteria, and second one viruses, third one fungi, and fourth one parasites. Then what is meant by bacteria? So bacteria, it is a prokaryote. So what is meant by prokaryote? Prokaryote means they have primitive nucleus. So primitive nucleus means there is no nucleolus and no nuclear membrane. Bacteria, it is a prokaryote, which means it has primitive nucleus. Primitive nucleus means there is no nucleolus and no nuclear membrane. So they are unicellular organisms. Bacteria, they are unicellular organisms, which means they are made up of single cells, except higher bacteria such as actinomycetes. Actinomycetes, it is a higher bacteria. So except these actinomycetes, remaining all are unicellular, which are made up of single cells. They are measured in micron or micrometer. Bacterial size is measured in micron or micrometer, and the bacterial cell wall is made up of peptidoglycan. It is a complex carbohydrate. Bacterial cell wall is made up of a peptidoglycan, and they grow on artificial culture media. Bacteria can be grown on artificial culture media like nutrient agar or blood agar or chocolate agar, like any of the culture media, they show the growth on artificial culture media. And how the bacteria divides? The bacteria divides by the process called binary fission. And study of bacteria is called as bacteriology. Then what is bacteria? Bacteria, it is a prokaryote, which means it has primitive nucleus. Primitive nucleus means there is no nucleolus and no nuclear membrane. They are unicellular organisms, except higher bacteria such as actinomycetes. Their cell wall is made up of peptidoglycan and their size is measured in micron or micrometer. They grow on artificial media and they divide by the process called binary fission. And study of bacteria is called as bacteriology. The next, viruses. Viruses, they are not prokaryotes nor eukaryotes. They are akaryotes, or they comes under organisms beyond cellular organization. So generally, organisms with the cellular organization, which means those who are having the cell structure, they are only classified as prokaryotes or eukaryotes. So viruses, they have no cell structure. So that's why they comes under organisms beyond cellular organization. Or we can consider them as akaryotes, which means absence of nucleus. There is no cell structure at all. So they are obligate intracellular infectious agents. Viruses are obligate intracellular infectious agents, which means strictly they grow inside of the living cells only, and they are infectious agents. They causes infections, and their size is measured in nanometers. Viral size is measured in the nanometer. So as you know, nanometer is smaller than the micrometer, which means viruses are smaller than the bacteria. Viruses, they contain either DNA or RNA as genetic material, but never both. They consist of either DNA or RNA as genetic material, but never both. If they consist of DNA as genetic material, they are called as DNA viruses. If they consist of RNA as genetic material, they are called as RNA viruses. And viruses cannot survive independently. They always depends upon the host cell. Here, who is host? Either human or animal. So here they always depends upon the host cell for their metabolism. And viruses cannot be grown on artificial media. They do not grow on artificial media. They require living cells. Okay, like they grow in the embryonated eggs, or otherwise in the animals, or in the tissue cultures. So they grow only on the living cells, and they require electron microscopy to see. So with the normal microscope, with the compound microscope, we cannot see them. So to visualize the viruses, we need electron microscope. Only with the help of electron microscope only, we can see the viruses. And study of viruses is called as virology. Next, fungi or fungi. So what is fungi? Fungi, it is an eukaryote. Then what is meant by eukaryote? Which means they have true nucleus. So true nucleus means there is nucleolus and a nuclear membrane, and they have well-developed cell structure. And if you see, fungal cell wall is made up of chitin. Fungal cell wall is made up of chitin, and their cytoplasmic membrane consists of sterols. And fungi, some are unicellular organisms and some are multicellular organisms. If you take a Cryptococcus neoformans, it is a unicellular organism. And like Aspergillus or Penicillium, they are multicellular organisms. They are measured in microns or micrometers. Their size is measured in microns or micrometer, and they reproduce either sexually or asexually or by both. Fungi, they reproduce either sexually or asexually, and they can be grown on artificial culture media. So mostly they grow on Sabrous dextrosugar media, and Sabrous dextrosugar with antibiotics and BHA, brain heart infusion occur. So they can be grown on artificial media, and the study of fungi is called as mycology. Next, parasites. Then what is meant by parasites? Parasites, they are the organisms which always depends upon the host. Parasites, they are the organisms which always depends upon the host, which means like they depends upon the host for their nutrition, survival, for their like life cycle, everything that depends upon the host without giving any benefit to the host, and always host suffers from the disease. So parasites always depends upon the host for their nutrition, survival, for everything that depends upon the host without giving any benefit to the host. They causes diseases in the host. See, uh, all parasites are not microscopic. So some of them are visible also. If you see, elements are macroscopic. So again, parasites, there are two types, protozoans and elements. So what is meant by protozoans? Like protozoans, they are unicellular eukaryotic organisms. Protozoans, they are unicellular eukaryotic organisms. And if you see elements are multicellular eukaryotic organisms. Protozoans are unicellular eukaryotic organisms and elements are multicellular eukaryotic organisms. And protozoans, they belongs to the kingdom Protista, and elements, they belong to the kingdom Animalia. And protozoans are microscopic structures, which means they can be seen under microscope. So if you see like entamoeba, histolytica, plasmodium, so all they are like a protozoans. So they can be seen under only microscope, and their size is measured in microns. Protozoans size is measured in microns, and the study of protozoans is called as protozoology. And if you see elements such as nematodes or trematodes such as Ascaris lumbricoids or Fasciola or like Ankylostoma duodenale, so they are multicellular eukaryotic organisms and they are macroscopic. So macroscopic means like without the microscope also they can be seen. Why? Because they have the size, their size is measured in millimeters. So as you know, millimeters, it is visible. So then why, even though they are visible, why we are studying as a part of like microbiology? Because like even though they are visible, they require the microscopic identification, especially their eggs or larvae require the microscopic identification. So like a study of elements is called as the elementology. Then what is the difference between protozoans and elements? Protozoans are unicellular eukaryotic organisms. Elements are multicellular eukaryotic organisms. And protozoans are microscopic structures. They can be seen under microscope and they're measured in microns. And the study of protozoans is called as protozoology. And the elements are macroscopic and their size is measured in millimeters. And the study of elements is called as elementology. And study of parasites is called as parasitology. And if you see, protozoology and elementology, they are two branches of parasitology. Next, immunology. So what is meant by immunity? So immunity means a protection. That is a resistance which is exhibited by the host against to the like pathogens is called as the immunity. So immunity means a protection. Immune means a protection. So then what is meant by immunology? Immunology, it is the study of immunity system or it deals with the protective mechanism. Then what we study in the immunology? So it includes the serology. So in the serology, what we do the tests, we detect the like products of immune reactions in the blood or serum, and it helps in the diagnosis of infectious diseases. Next, scope of medical microbiology. What is the scope of medical microbiology? Why you have to study the medical microbiology? So medical microbiology helps you to understand and identify the pathogens. It helps you for understanding the and identification of pathogens. Means like you will understand what is bacteria, what is virus, and what is fungi, and you can identify the like various forms of the like microorganisms or various forms of the pathogens. And it helps you for confirmation of diagnosis. So it helps you for confirmation of diagnosis. So suppose if you come to know the like diagnosis, it helps in the treatment strategies for specific diseases, like what type of antibiotic to be given, what type of antiviral agent to be given. That you'll come to know with the help of medical microbiology if you study medical microbiology. And then it helps you to detect the newer pathogens. Medical microbiology helps to detect the newer pathogens, and it helps to determine antimicrobial susceptibility patterns. So various like bacteria, they show the susceptibility patterns, different type of susceptibility patterns also. Some bacteria are resistant to the like antibiotics and some are susceptible to the antibiotics. So you'll come to know the susceptibility patterns of various pathogens so that you can plan for the treatment. And it helps you like for development of like vaccines and immunotherapeutic agents. So vaccines which prevent the like diseases, which prevent the like clinical attacks, and it helps for development of newer antimicrobial agents or new type of antibiotics, and it helps you to understand the mode of transmission, and it helps you to find out the source of infection. So you'll come to know like how one infection is transmitting from one to the another one. So some of the infections are transmitted by like inhalation, or some of them are transmitted by ingestion, and some of them are transmitted by contact. So you will come to know like how the infection is transmitting from one to the another one, and you can able to find out the source of the infections, so that you can take the like preventive measures. And it also helps you to detect the asymptomatic infections. Asymptomatic infection means like some people like they will be having the like infection, but they do not have the symptoms. There is no clinical manifestations. And even it helps you to identify the like chronic carriers. And next, like you can distinguish between recent and past infections, and it helps you to monitor the response to the treatment. So whenever we give an antibiotic or antiviral agents, like how the host immunity system is reacting against that one, or how host body is reacting against the treatment also will come to know. And you'll understand the role of body immune system. So how immunity system is reacting against to the pathogens, like what type of antibodies are produced, like how long that antibodies like can be demonstrable in the like serum, that you will come to know. And maintaining of general hygienic conditions, that is based upon principles of microbiology, medical microbiology, and disposal of hospital waste, that is also one of the principle of the microbiology. This is the end of this session. So if you see summary, what is microbiology? It is a branch of science which deals with the study of microscopic organisms, which means which can be seen under only microscope. So bacteria, viruses, fungi, and parasites, they are microscopic forms. And if you see bacteria, fungi, and parasites, they are cellular forms, and viruses are acellular forms. And knowledge in microbiology helps you in identification of pathogens, diagnosis, and treatment of such infections. Thank you.